So the cardiac cycle is the sequence of events that happen from one heartbeat to the next, or one complete cycle of the heart pushing blood through the heart. And what you learned last time and yesterday and in lab about the anatomy of the heart is really, really important to understand this. So I'm really glad that we had lab right before this, and so you're pretty strong now with the anatomy of the heart. But everything that happens in one heartbeat, how long is a heartbeat? Not long. That's a good answer. It depends. Like a second. Pretty short amount of time. Everything that happens in between heartbeats, that's a cardiac cycle. And there are a couple of definitions that we need to know. And also pronunciations. These are weird pronunciations. The first one is called systole. Systole. But that's how you pronounce it. Systole. And when a part of the heart or a chamber of the heart is in systole, that's when it's contracted. That's when that chamber is contracting. So if we talk about atrial systole, that's when the atria are both contracted. If one of the chambers is in diastole, that's how you say it. It's not how you read it, that's how you say it. Diastole, that's when that part of the heart is relaxing. So each chamber on its own will contract and relax, contract and relax. The atria go together, or they're supposed to. The ventricles go together. So you should have both atria in systole and then in diastole. And the ventricles are mostly are doing the opposite. There's a little brief amount of time when all the chambers are relaxed, when all the chambers are in diastole. But for a large amount of time, when the atria are contracting, the ventricles are relaxing. When the ventricles are contracting, the atria mostly are relaxing, generally. So don't get those backwards. Here's how I remember it. Diastole is like diameter, and when the chamber is in diastole, it has a bigger diameter and it's relaxed. That's how I remember it. You may come up with a different way. So this part of the class is assuming that you've got the naming of the heart chambers down. And in these pictures, they're just abbreviating them RA, LA, LV, RV. So right, left, atria, ventricles. And it's kind of also assuming that you remember the path that blood takes. So I'm going to stop here for a minute, and you might, if you've got pictures, you might want to take a pencil and trace it with me. The path that blood takes through the heart. And I'll start at the usual place, which is superior and inferior vena cava, coming into the right atrium, going down through the tricuspid valve, or the right AV valve, down to the right ventricle, up through the pulmonary valve. What's this? Pulmonary trunk. One thing I corrected on labs yesterday is the pulmonary trunk is before it branches, and then it goes out pulmonary arteries. Then it goes to the pulmonary circulation. Then it comes back through pulmonary veins, which we don't see on here. It comes back into the left atrium, down through the mitral valve, or bicuspid, or left AV, into the left ventricle, up through this valve, aortic valve, into the aorta, and then where? Yeah, that's right, into the systemic circulation. It leaves red, it comes back blue. On models it looks that way. And then it comes back. So you might want to practice. It's kind of fun, it's kind of relaxing. But don't just draw it, remember the parts that it's going through. So, now that we have reviewed the circulation, we're going to start at the right atrium. And you have to, it's a cycle, but you have to start somewhere. You have to pick a place to start. So we're starting at the point when the atria are relaxed, and whatever is happening in this atrium will also be happening in this atrium. So we're starting with blood coming into the atria. They're relaxed at this point. They're in atrial systole, atrial systole is when they're contracting. We're starting with them. They've already got blood in there. They're contracting. And they're pushing it down into the ventricles because they're contracting. Why does it go into the ventricles and not back out the other way? Valves. What valves? They contract and they push it, they open the valves and they push it down this way. Why doesn't it go backward? You actually don't know that yet. But that's something to think about. We'll talk about that in a little while. So they contract, atrial systole, they push blood down into the ventricles. What should the ventricles be like if blood is going to get pushed in them? They are supposed to be relaxed at that point. So ventricles are in diastole. The valves open when the atria contract. That's what makes them open. They otherwise would be closed. Does that seem okay so far? Okay. Let you write the blanks. And notice on here, whoa, the ventricles are now totally clamped down. That makes sense because they're contracting. Notice the walls have gotten a lot thicker because the muscle fibers are shortening. So it's like clamp this down, squeeze blood up when the ventricles are in systole. So when the ventricles start contracting, they've already gotten blood from the atria pushed in. They start contracting. And that contraction makes those valves shut that the blood just came through. The contraction of the ventricles shuts the AV valves. It's like pushing the door backward. So the blood can't go back that way. That's a good thing. Where does it go instead when the ventricles contract? Up and through the pulmonary trunk out of the right side, through the aorta out of the left side. So blood generally is going down, up, down, up. Down during the atrial systole, up during the ventricular systole. When the ventricles contract, that's what opens these valves. They open out. Any of you work like with cows or herd animals, where you have to make them go through gates and not come back where they came from? You ever done that? You make the gates open only one way, and then they kind of get, like you can kind of make them get in a panic, and then they'll go that way. And you get some of them going that way, but they can't come back because the gate only opens one way. Blood going through the heart is like that. So ventricles contract, these valves, the pulmonary and the aortic valve open, blood gets pushed to its next location. That's ventricular systole. What's happening to the atria right now?
They're relaxed.
They're relaxed, they're in diastole, and what else is happening while they're relaxed?
Blood is going in.
Blood's still coming in. So you can kind of assume all the time that all the time blood is coming back into the atria. It's easier for it to come in when they're relaxed. Seem okay? So we've had blood come into the atria, into the ventricles, now gone out of the ventricles. And that happens in about a second, down to 0.5 seconds, depending on how fast your heart is beating. And when we listen to our heartbeat, we're not listening to the heartbeat, we're listening to the valve slam. So if you can remember that, that would be useful. You remember that onomatopoeia concept, that the name of the word sounds like the thing? So it's loved up, loved up, loved up. We'll do some listening later in lab, listen to our heartbeats. So what are the lub and the dup sounds? Does that say that? It says that on there. Which valves close first?
The atrium, right?
Atrioventricular valves.
Yeah.
That's the lub. These are the dup. Why did these close before these?
So that blood can go.
That's good that it happens that way. So blood goes up. We want blood to go only forward and not back. The reason that these close before these is that the ventricles contract from the bottom up, which is awesome. We'll talk about why that is. They contract from the bottom up. So blood is first pushing here, then pushing here, shutting these, opening this. When do the pulmonary valve and the aortic valves actually close? What makes them close? Or what makes them stop being open? What'd you say? When there's no more blood flowing through? You know when they close? When the pressure in here is lower than the pressure out here. That's when they close. When we push so many cows through the gate that they start like trying to come backward, but closes it. So the AV valves close early during ventricular contraction or ventricular systole. The semilunar valves up here, they close late. They close at the end of the ventricular contraction. Loved up, loved up, loved up. What happens between loved up and then the space before you have another one? What's in diastole? There's actually a lot of stuff that's happening in that space. So if you think about your heartbeat, loved up, loved up, loved up. Most of it is not valves closing. So the dup is these closing. What's happening right after that in the atria? They're filling up. They're always filling up. That's a good answer. They're filling up. When they fill up enough, when they have filled up a reasonable amount, they're supposed to be contracting. Push blood out. Does that make a sound yet? No. Remember, the sound is not contracting. The sound is the valve shutting. So these are filling up. They'll start contracting. Push the valve open. That starts filling the ventricle. We don't get a lub until the ventricles contract and push that valve shut. And open this one, push blood through. When this one shuts, that's the dup. While I'm thinking about it, there's a review video for this whole process. It's like 2 minutes long. It's great. I might show it at the end of today, but it's also on Blackboard, and it shows the path of blood. It shows, you can sit there and say, loved up, loved up. See everything that's happening. It's really good. So one thing that I will not be teaching you in lab is how to diagnose people's heart problems with a stethoscope. That's very that requires more training than I have. We can hear lub dup. If you ever get good enough at this and you hear swooshing, gurgling sounds, that's indicative of a heart murmur. And a heart murmur is when the valves are not operating properly and you do have backward swishing through the valves. And if you know what you're doing, you can hear it if you listen at the right place. If you listen at the right place. That's what heart murmur is. So that's the cardiac cycle. The lub, the dup, and the in-between, systole, diastole, the path that blood takes. What I'm going to talk about next is how does it actually contract? What makes it contract in rhythm? And this is especially cool, I think. So before we start on this, know that it's called the heart's intrinsic, that's like in itself, on its own, intrinsic conduction system. And it's operating whereby the heart muscle itself is making itself contract. Our brain can change the speed at which it contracts, but it will contract on its own. If you've ever seen one of those terrible movies where or like video games sometimes where like somebody is holding a beating heart in their hand, that's not, it will cause itself to beat as long as it has oxygen and ion flows to do so. It's a real thing. It will keep beating until it runs out of oxygen, glucose, and correct ions. But yes, it can beat on its own, but it's crucial that the conduction system works well so that your heart is contracting not just in the right interval, but with the right spatial organization. So there's timing to it, there's spatial organization to it, so that atria contract, and then ventricles contract, and those things are happening at the correct timing apart from each other. And we're going to be looking at, in this picture, the yellow stuff. The yellow stuff. This material for me is lecture topic, not lab. So we don't have this on our lab list, but it is an important lecture topic. And there are certain anatomical features of the heart that are listed on here that you pretty much need to know. Mainly the SA node, that stands for sinoatrial, AV node, atrioventricular, and these are the places in the heart that can generate and conduct electricity. So there's a little blob up here, SA node, there's another blob down here, AV node, and then that's connected to branching pathways that go down. What do we call this part?
Apex.
Apex. The one everybody knows. Go down toward the apex and then come back up. You might want to write on here some numbers for how this proceeds. So you see this set of arrows up here? That's number 1. That's the first thing that happens in the cardiac cycle. We get electrical signals coming from the SA node and they spread through these two chambers, which are what?
Atria.
The atria. That's the first electrical signal. The second electrical event is signals reaching AV node. That happens second. So this would be number 2. Number 3 is the signals reaching the apex. And then number 4 are signals going up the ventricular walls. Those of you that later have exercise phys or more in-depth heart physiology will go into more detail than I am here. But 1, 2, 3, notice I did that really fast. 3, 4. That's going to be the And as those signals travel through, in that order, through those pathways, they are depolarizing, or those depolarizations are causing muscle contraction. The way this happens is basically the same stuff that you learned in AMP1 about how generally muscle contraction works. You have nervous stimulation, you have various ion flows, troponin, myosin, you remember all that? Generally the same in heart muscle. So you get signal. First you get electrical signal, and then shortly thereafter, then you get the muscle contraction. So if we want atria to contract before ventricles, the spatial organization is important. So is the timing. And when I learned about this, I thought this is one of the most awesome things. It's really cool. So here's generally how it works. We start at the SA node, sinoatrial. This is the main pacemaker in the heart. This is the heart's own pacemaker. Are these out of order? I think these are out of order possibly in your packet. Maybe not. Maybe not. Good so far? Okay. Starts here. This is the pacemaker. This is the heart's own dominant rhythm setting tissue, the SA node. It starts the electrical process. And it sends a signal through the atria, and that signal travels kind of slowly. So it starts over here on the right side. It can jump over to the left side through a particular area right here. Jumps over to the left side, but it moves kind of from top to bottom. I'm concerned now. From top to bottom, from the top of the atria toward the bottom. And that's great because that means that the atria will contract just slightly, lagging behind the signal from top to bottom. That's great. It's pushing blood from top to bottom down to the ventricles. So kind of a slow signal traveling down. The contraction is coming right behind it, pushing blood down, which makes sense. Okay, so once the electrical signal makes it from the SA node down here to the AV node, this is named atrioventricular because of where it is. Atrioventricular, AV node. You notice that's in yellow, and then all this stuff is in yellow. The yellow pathways are fast. The yellow pathways are like a highway. This is like walking through the grass from place to place. This is like driving quickly from place to place on a highway. There's one other important thing you need to know to understand how this works. Outside of here, where you see this white stuff, that's like electrical insulation. It acts like electrical insulation. So these pathways, AV bundle branches, coming down to here, they're conducting the signal, but they're not transmitting it to the muscle wall. It's just signal moving. It can't get to the muscle wall until, guess when? It gets down here. So this signal, you get a slow signal coming down here that's causing atria to contract from top down. It reaches here, shoom, quickly goes down here, but it didn't cause contraction along the way. When it gets down here, there's no more insulation, or less. And now, as it travels back up, guess what? These contract from the bottom up. That is so great. That's so great. We get blood not only contracting the chambers are contracting in rhythm, but they're also contracting in a spatial organization. Atria from top to bottom, ventricles from bottom to top. Questions so far? Not only does your heart beat on its own, it does so in a very sophisticated way. So, once the signal gets down to the apex of the heart, contraction starts at the bottom, and also the muscle layers of the heart are arranged in a twisting way in the ventricular walls. So contraction starts at the bottom, and it's like wringing out a rag from the bottom up. So pushing blood from the ventricle at the apex upward through what valves? Mini quiz.
So,
What?
that's the valves.
Semilunar valves, yeah. So the aortic and the pulmonary valves. From ventricles up. Keep coming back to this slide. This slide is critically important. And you might want to, print it out big or practice drawing on it. Atria contract from top to bottom, starting with the SA node. Ventricles contract from bottom to top, even though the signal to do that originates up here. That's because it travels quickly down to the apex, but the muscle walls don't start getting stimulated until it gets down here, and then they contract bottom up. That gives time for the atria to finish contracting. Blood is going into the ventricles, and then they contract from bottom up. Key terms: SA node, AV node, bundle branches, and then the other ones that you sort of want to recall are Purkinje fibers. The Purkinje fibers are the ones that are causing depolarization in the ventricles. Why do you think they're named that? Why such a weird name? Someone's name who first found them and wrote it down. Purkinje.
Can you spell that?
Can I spell it?
Yeah, Purkinje.
Yeah, P-U-R. I have to look. P-U-R-K-I-N-J-E. Yeah, Purkinje. I don't know anything about Purkinje, but people learn the fibers that are named after them. This slide is not in your packet because I realized today, oh my goodness, why is this slide not in your packet? So I put it in my slides. It's not in your slides. So this information, you'll need to copy it down, all the words, onto your slides. But what this is about is, yeah, the heart can beat on its own, starting at the SA node. I'll let you copy and then I'll talk about it. Because I know you won't listen while you're copying. This is why I do the notes the way I do. I hate spending class time on copying words. Not a great use of time. Okay. Go. Right, quickly. Okay. So what you're looking at here, here's a heart. The yellow segments are what we just talked about on the previous slide, the intrinsic conduction system. And we're looking at connections between the nervous system and the heart's conduction system. Before I explain this, I need to go back again to that previous slide, 2 slides before, and tell you something about the relationship between the SA node and the AV node. Okay. So the SA node is really the dominant pace setter, pacemaker. And it sets a heartbeat, this is a estimate, of about 80 beats per minute. That's its natural pace. So if it's just all on its own and nothing else is sending the heart information, your natural heartbeat is 80. If something happens to the SA node, like you have a heart attack and it damages this region up here, something happens, the AV node is also a pacemaker, but it's just kind of usually overridden by the SA node. So the AV node itself can keep the ventricles beating. It can keep the ventricles beating, and I think it's about 40 beats per minute, which is not very efficient, barely enough. But if something happens to the SA node, AV node doesn't get overridden, and it will provide a heart rate at about 40. Usually, your heart rate is neither 80 nor 40 because your nervous system can provide input to how fast your heart beats. Mostly through the parasympathetic division and the sympathetic division, like we talked about at first day of class. So your brain will be sensing things like, what is the blood pressure in your aorta? And is it dangerous right now? If so, let's slow down the heartbeat. There are also sensory neurons that come from the arteries up in your head, in the carotids, that provide input and help your brain decide whether your heart rate needs to be faster or slower. Things like if you see something scary, hear something scary, smell something scary, those will also be working through your sympathetic nervous system to cause you to go into flight or fight response and do what to your heart rate? Increase. If you're just having a normal day and you're not freaked out and nothing is scaring you, you're not in fight or flight, your parasympathetic division will normally slow heart rate, like in your average person, to about 70. So that's why a normal healthy heart rate in a person at rest who doesn't have an underlying condition will be closer to 70, because your parasympathetic nervous system is slightly slowing down your SA node activation. So problems with the nervous system and problems with the heart the heart tissue itself can make pretty big changes in your heart rate. We're also going to measure heart rate in lab next week, but not from scaring you, I hope. It'll be from like exercise. All right, so I want to do a little brief intro to EKGs. It stands for electrocardiogram. Electrocardiogram. EKG. And these are measuring electricity. These are measuring the electrical signals. They're not measurements of contraction, but they imply if you get this signal, you're probably going to have a contraction proportional to it right after that. But they're measuring electricity. What's EEG? It's something different. Electro something gram. What do those measure? Electricity where? I bet you didn't do these in lab. It's brain electricity. So EEG, electroencephalogram, brain activity. EKG is heart electrical activity. And these are measuring, you can measure on the outside of the body the electrical signals that are coming from the heart. And they have this particular shape. And I would kind of like you to be able to draw a reasonable EKG shape, it is something like this: a small bump, a big bump, a small dip, back up, a medium bump, kind of like that. And we'll do a lot more with these in lab. But when you do this, when you're measuring these, this level right here is the baseline. That's the baseline. And that would represent what's the electrical state of your heart in between the depolarizations. So next week when we're in lab, it'll say, do you have a flat baseline? That's what it means. You got to start with a flat baseline before you get a good measurement. And as you look at these pictures, you'll see there's yellow. So we've got the 4 chambers of the heart. There are yellow waves and there are green waves. The yellow, do I have that on here? The yellow is depolarization. That's the initiating electrical signal. So you see yellow starts here. It all starts slightly quicker on the right side than the left. The atria depolarize, and the ventricles depolarize. And this shows you the direction too, which I like. Top down, bottom up. The green is the reset, the repolarization, coming back to baseline. So when you see these EKG waves, it's showing you you've got a small depolarization, and then you've got a big depolarization. The big one is for the ventricles, because they're bigger, they have more electrical signal. So this bump represents atria depolarizing. This big thing represents ventricles depolarizing. What are ventricles in systole or diastole there? Up here, they would be in diastole because they're relaxed. Down here, the actual contraction wave would be following the yellow. So they might be starting to be in systole down here. At that part, it's going to follow the yellow wave. And then the green is the reset. This second bump that you see, that's the ventricles repolarizing, resetting. That's represented by the green, which is probably on the next picture. Yeah. So you've got ventricles depolarized all the way. They would also, right after that, contract from the bottom up. They also reset or repolarize from the bottom up. That's the green wave. So that bump, the T wave, it's called T wave. We'll review that again in lab. That's the ventricles repolarizing. This bump is for the atria depolarizing. This big peak is for ventricles depolarizing. The atria also repolarize, but it's hidden by this. It's so minor of a contribution that it's hidden by the ventricle depolarization. So if you were to draw a chain of these, or we will see a chain of these, this is not perfect, but there's a lot of bad versions out there, like on coffee mugs and fundraiser T-shirts. Like there's something wrong with that person. So what we're going to be doing, we'll stop here. What we're going to be doing in lab next week is measuring those, learning more about them, looking at abnormal ones. You need to bring these notes with you next week. Tyler, can you push that?